Above powerful thunderstorms, enormous electrical flashes can appear where the atmosphere is already approaching the edge of space. These events occur far above ordinary weather, across atmospheric layers most people never associate with lightning. Above storms, conventional lightning moves through clouds or toward ground, but other electrical disturbances illuminate the sky overhead, across altitude. At those heights, air density, pressure, and electrical breakdown behave very differently from conditions near Earth's surface. Thin air allows enormous luminous structures to develop from surprisingly small amounts of excited gas, above active storms. Excited nitrogen produces much of the characteristic red light seen in high-altitude sprites. Structure identifies mesospheric response. Most transient luminous events are faint, brief, and easiest to detect with sensitive cameras after darkness falls. A complete event may unfold within milliseconds, disappearing before an observer can consciously process its shape. Across altitude, high-speed imaging transforms one mysterious flash into a sequence of rapidly developing electrical structures. During brief flashes, the storm below can trigger light tens of kilometers above its cloud tops without a continuous visible bolt. Understanding the upper flash begins with how collisions inside thunderstorms separate electrical charge. This signal resolves storm charge. Violent collisions among ice and water particles help organize charge throughout a growing storm. Contrast tracks mesospheric response. Storm dynamics create large reservoirs of opposite charge separated across several kilometers of cloud. Boundary locates lightning coupling. As charge separation increases, the surrounding electric field strengthens throughout the cloud and nearby atmosphere during brief flashes. Lightning begins when electrical breakdown creates conductive channels through air that was previously insulating. Transition compares electrical breakdown.
Some sprite-producing flashes transfer positive charge from the cloud toward Earth in exceptionally powerful strokes. After lightning,
that rapidly changing field can push thin upper air beyond its local breakdown threshold. Emission exposes mesospheric response.
Red sprites occupy the mesosphere and lower ionosphere, dramatically higher than thunderclouds or commercial aircraft, within thin air.
Their scale becomes clearer when placed above aircraft routes and below most orbiting spacecraft. Evidence frames sprite formation. A sprite can illuminate a region far larger than the storm feature that appears to trigger it. Enormous size does not imply long duration, because most visible sprite structure develops almost instantly, during brief flashes. Some sprites follow their parent stroke almost immediately, while others appear after a measurable delay, above active storms.
Both rapid charge removal and continuing current can influence whether the upper atmosphere reaches breakdown, within thin air.
Researchers use charge moment change to describe how strongly lightning alters the electric field above a storm.
Charge moment changes strongly increase the probability that a lightning flash produces a sprite across wide regions. Even powerful positive strokes do not guarantee sprites, because atmospheric conductivity and storm geometry also matter, within plasma. Sprite initiation depends on several interacting conditions, rather than one simple lightning measurement. This boundary maps global occurrence. Nighttime conditions favor optical detection and change the electrical response of thin mesospheric air above active thunderstorms.
Solar radiation alters electron density, affecting how electric fields build and dissipate above storms. Contrast tracks charge transfer.
Many events are observed at night because their faint emissions would disappear against bright daylight across wide regions.
Observers often photograph sprites from distant clear skies rather than standing beneath the parent thunderstorm during brief flashes.
Distance can improve visibility by exposing the entire region above a storm's cloud shield. Transition compares global occurrence.
Sensitive imaging captures emissions too brief and dim for ordinary cameras under most conditions. Network supports upper fields.
Precise timing links each upper atmospheric event with candidate lightning activity inside the storm below, after powerful lightning. These matched observations establish the connection between sprites and powerful lightning charge transfers. This channel distinguishes storm geometry. Sprites occur worldwide wherever sufficiently energetic thunderstorms interact with favorable upper atmospheric conditions. They are not limited to tropical convection, although storm structures and triggering patterns can differ above active storms. Many sprites resemble luminous jellyfish, with a bright body suspended above long descending tendrils. Channel tests ionospheric change. Individual streamers propagate through localized electric fields and build the sprite's intricate lower structure. Evidence frames radio effects. Ionization grows around moving streamer tips, producing visible channels that divide repeatedly. This observed response measures ionospheric change. Different streamer polarities can develop in opposite directions within the same enormous event. This observation reveals halo development. Other events appear as simple luminous columns with little branching visible at ordinary video speed, within thin air. Sprites frequently occur in clusters distributed across a broad region above the parent storm. Observation records ionospheric change.
Successive sprites can appear to dance across the sky as lightning activity moves through the storm, across altitude.
Wind shear and tilted storm charge can shift sprites away from the parent ground stroke during brief flashes. The upper discharge may not sit directly above the point where lightning reaches the ground, above active storms.
A broad sprite halo sometimes appears before or without bright streamer development. This synchronized geometry reveals halo development.
Halos arise from diffuse electrical excitation rather than the narrow channels defining mature sprite streamers after powerful lightning.
Under suitable fields, a halo can transition into structured streamers that grow downward rapidly. Boundary locates ionospheric change.
Microsecond imaging reveals a developmental sequence hidden inside the brief visible flash. This measured sequence links streamer growth. Bright streamers can travel extraordinarily fast through the thin upper atmosphere. This field explains streamer growth above storms. The event extinguishes once local fields can no longer sustain ionization and energetic electron collisions within thin air. Although the light vanishes quickly, the upper atmosphere can remain electrically disturbed afterward. This sequence connects plasma motion.
Sprites and related events can produce measurable changes in very low frequency radio propagation. Emission exposes streamer growth.
Remote radio measurements help detect electrical disturbances even when clouds or distance block optical observations during brief flashes. Combining cameras, radio sensors, and lightning networks reveals how thunderstorms couple electrically with the upper atmosphere above them. Blue jets begin near storm tops and propagate upward through the stratosphere, rather than appearing suddenly far above. Unlike sprites, blue jets develop upward from the thunderstorm itself. This signal explains jet development within thin air. Typical blue jets terminate well below the highest sprite structures. This transition compares event rarity within thin air. Different colors reflect atmospheric density, composition, and the energies exciting nitrogen molecules. This mapped profile connects charge transport. Blue jet emissions arise from excited and ionized nitrogen within denser stratospheric air above thunderstorm cloud tops. Ion streamers combine into the luminous structure recorded by sensitive high-speed cameras, revealing jet development frame by frame. Observed blue jets follow extremely brief electrical activity embedded within the cloud. This profile links plasma structure. Storm charge arrangement determines whether breakdown remains internal or escapes toward the stratosphere. This pattern tests global circuitry. Some upward discharges grow beyond ordinary blue jet altitudes and enter a different category. Response measures cloud escape. Gigantic jets can create a direct electrical connection between thunderstorms and the lower ionosphere. Profile connects jet development. Their upper branches reach approximately the same height as the base of the ionosphere. Pattern distinguishes event rarity.
A single gigantic jet can extend many times higher than the storm producing it. Contrast tracks plasma structure. High-speed observations show that gigantic jets can advance through repeated leader steps and streamer bursts across altitude. Different plasma structures coexist within the same upward discharge. This response constrains cloud escape within the upper atmosphere. Most attempted branches terminate before reaching the ionosphere. This profile links jet development within the upper atmosphere. The discharge changes structure as air density and conductivity vary with altitude. This mapped evidence frames charge transport.
Observed gigantic jets can transfer substantial electrical charge directly upward. This response measures global circuitry within thin air.
Gigantic jets represent one visible pathway connecting storm electricity with the planet-wide atmospheric circuit across wide regions. These jets are uncommon relative to the enormous number of lightning flashes occurring worldwide. Trace constrains global circuitry. Limited observations still prevent a complete global climatology of gigantic jets, especially across remote tropical oceans and continents. ELVs are enormous, faint rings produced when a lightning electromagnetic pulse strikes the lower ionosphere, within thin air. An ELVE can spread across a region hundreds of kilometers wide within a fraction of a millisecond. The parent lightning does not need to send a physical channel into the ring. Measurement maps historical discovery.
Electromagnetic energy briefly excites the lower ionosphere and creates the expanding optical emission. This observation reveals historical discovery.
Its geometry reflects the outward-moving electromagnetic pulse intersecting a thin atmospheric layer. This timing exposes sensor fusion. Most ELVs vanish within about a millisecond, making them among the fastest transient luminous events, within thin air. Different event families occupy distinct timescales despite all being associated with thunderstorms. This synchronized boundary locates optical detection. Photometers aboard spacecraft are especially effective at detecting ELVs. This sequence links sensor fusion within thin air. Viewing from above removes cloud obstruction and reveals the event's true horizontal scale. This contrast frames ELV expansion. Repeated orbital observations help estimate occurrence rates beyond what ground cameras can measure. This boundary maps historical discovery.
The first confirmed Sprite recording appeared accidentally during an unrelated nighttime camera test in July 1989.
Repeated viewing revealed that the strange flash was real rather than a camera artifact. Contrast tracks event timing.
Shuttle observations soon provided more examples and confirmed that the phenomenon extended across large storm systems, across altitude. Different perspectives revealed structures that looked confusing or incomplete from the ground. This brief sequence links event timing. Coordinated observations reconstruct altitude, size, and timing more accurately than any single camera. Researchers combine optical imagery with storm structure and electrical measurements to identify triggering conditions. Network supports sensor fusion.
Millisecond synchronization separates plausible parent flashes from unrelated lightning occurring nearby. This structure supports ELV expansion above storms.
Raster cameras expose plasma motion that ordinary video compresses into one featureless flash. This channel distinguishes optical detection. Spectroscopy identifies which molecular transitions generate each event's characteristic light. This pattern distinguishes historical discovery within thin air. Color and spectrum provide remote clues about otherwise inaccessible upper atmospheric conditions. This map structure identifies event timing. Space-based instruments now watch thunderstorms from above, where cloud tops cannot hide upward electrical activity after lightning.
ASIM combines imaging, fast light measurements, and high-energy detectors during repeated passes over active storms, above storms.
Orbital measurements captured blue jets beginning with extremely brief electrical activity inside cloud tops. Measurement maps forecast skill. Multi-channel observations distinguish genuine events from reflections, cosmic rays, and ordinary lightning glare. Field resolves ionospheric response.
From orbit, ELVs appear as complete rings rather than partial arcs hidden near the horizon, above active storms.
Astronaut photography complements automated instruments by targeting storms selected from forecasts and real-time observation within thin air.
Successful observations require predicting where intense storms will intersect a spacecraft's narrow viewing window. Observation records remaining uncertainty. Skilled citizen observers now contribute valuable images, timestamps, and storm locations to scientific databases. Timing clarifies sensor coordination. Multiple viewpoints can estimate altitude and structure, even when no dedicated instrument observed the event, during brief flashes.
Reconstructions reveal how upward leaders develop inside storms before escaping into the atmosphere. Profile connects ionospheric response.
Scientists investigate whether transient luminous events measurably alter chemistry throughout the middle atmosphere and lower ionosphere locally.
Individual events are dramatic visually, but their global chemical importance remains uncertain. This mapped emission exposes ionospheric response.
Electrical disturbances can outlast visible light and temporarily modify the lower ionosphere. This resolved trace constrains chemical effects.
Background atmospheric waves may influence where breakdown begins and how sprite structures develop. This geometry records remaining uncertainty. Upper atmosphere conditions change continuously, complicating prediction from lightning measurements alone. This field explains orbital monitoring above storms.
Future systems may combine charge transfer, storm geometry and ionospheric conditions to forecast events. Network supports ionospheric response.
Many questions were made about why apparently similar thunderstorms produce entirely different upper atmospheric discharges. A mission exposes orbital monitoring. The field has moved from disputed sightings toward detailed physics, yet important parts of the system remain unexplained.
Thunderstorms do not end at the cloud top. Their electrical influence can reach the boundary between atmosphere and space.
One New Guinea songbird carries powerful poison through its skin and feathers. Hooded pitahui lives only within New Guinea's forested landscapes. Dense rainforest conceals a medium-sized bird with striking black and red plumage.
Its contrasting colors make the species immediately recognizable.
Bold coloration may help advertise an unpleasant or dangerous defense.
Potential predators could learn to avoid the bird after negative encounters.
Scientists discovered the toxin while conducting ordinary bird research.
Handling the bird produced unexpected burning and numb sensations. Those sensations suggested a biologically active chemical on the plumage. Field observations prompted chemical testing of feathers and tissues. Researchers compared toxin levels across different body tissues. Chemical separation isolated a potent steroidal alkaloid. The compound belonged to the batrachotoxin family.
Previously, these toxins were famous from poisonous frogs across another continent.
Unrelated animals had acquired remarkably similar chemical defenses.
The similarity pointed toward environmental acquisition rather than shared ancestry.
Diet became the leading explanation for how the bird obtained poison. The original dietary source was not immediately known. Toxin levels varied among birds collected in different places. Not every individual carried the same chemical concentration.
Batrachotoxin targets voltage-gated sodium channels in nerves and muscles.
Sodium channels normally open briefly during electrical activity. Batrachotoxin stabilizes channels in an open state.
Continuous sodium entry disrupts the membrane's electrical balance.
Affected cells cannot restore normal excitability. Muscle control also depends on properly timed sodium currents.
Cardiac electrical activity can also be disrupted.
The toxin acts by changing channel behavior rather than blocking the pore. Its chemistry allows close interaction with channel proteins. Structural studies reveal multiple contacts between toxin and channel. Binding favors an abnormal open configuration.
Many natural toxins target these essential electrical gates.
Batrachotoxin is distinctive because it drives persistent activation.
A painful encounter can teach avoidance without requiring ingestion. Learned avoidance could make small toxin quantities effective. Poison adds another protective layer to ordinary bird defenses. External tissues contain the highest measured toxin concentrations. Surface placement improves contact with attackers or parasites. The bird effectively wears a chemical defense. Toxic compounds occur directly within the plumage. Contact can deliver an immediate, unpleasant warning.
Researchers reported tingling, burning, and numbness after handling birds.
Those reports revealed toxicity before full chemical identification. Skin and feathers consistently held the strongest concentrations. Internal distribution appears uneven rather than uniform. The bird must transport dietary toxin without poisoning itself. Toxin is ultimately deposited into external tissues. New plumage may incorporate compounds acquired from food.
Molting may change the bird's total toxin reservoir. Toxic plumage may also affect feather parasites.
Anti-parasite benefits remain plausible but incompletely measured.
Loose feathers could reinforce predator avoidance.
Toxic feathers might influence the nest environment.
Whether chicks receive meaningful protection remains uncertain.
Toxin accumulation may depend on diet after hatching. Pitohies eat many arthropods rather than one exclusive prey.
Researchers searched stomach contents for a toxin-bearing source.
One little studied beetle group produced a compelling clue. The dietary trail pointed toward choricine beetles. Choricine beetles contain high levels of batrachotoxin compounds. Field samples confirmed that pitohies eat these beetles.
Diet offers a plausible route for toxin acquisition.
The compound must escape normal metabolic destruction. Unknown transport mechanisms likely move toxin into the body. Selective distribution suggests regulated sequestration. Geographic variation may reflect differences in available prey. Dietary exposure could explain variable toxin loads. Seasonal prey changes may alter chemical defense. Direct seasonal tracking remains limited.
chorocene may not be the only source.
The ultimate producer of batrachotoxin remains unknown. Scientists have not proven where the food web first creates the molecule. Chemical presence does not demonstrate biosynthesis.
Tracing labeled precursors could test where synthesis occurs.
Metabolic tracing could distinguish production from sequestration. Structural continuity would support direct dietary transfer. Chemical fingerprints may connect predator and prey. Shared compounds strengthen the food chain hypothesis. Acquiring poison creates a second problem: avoiding self-intoxication.
The bird's own sodium channels should be vulnerable to batrachotoxin.
A simple target site mutation once seemed like the obvious defense. Recent evidence does not support an easy mutation-based explanation.
Resistance may instead involve several protective mechanisms.
Carrier proteins could reduce exposure of sensitive tissues.
Rapid sequestration may keep circulating concentrations low.
Compartmentalization could isolate toxin from vulnerable channels.
Tissue distribution may provide part of the protection.
Metabolic transformation may also contribute, but evidence remains sparse.
Any detoxification system must preserve useful poison in external tissues. Self-protection and toxin storage must operate together.
Population comparisons could reveal resistance-related genes.
Genomic studies can identify candidate protective pathways. Resistance may be distributed across multiple systems.
Functional tests are needed to confirm protection.
Candidate mechanisms must change measurable toxin sensitivity.
Poison frogs face the same self-resistance challenge.
Similar chemical defenses need not use identical protection. The bird's resistance mechanism remains a major unsolved problem. The complete pathway from prey to plumage is still incomplete.
Chemical defense can reduce the value of attacking the bird.
Taste or contact may create rapid avoidance.
Predators can associate plumage with an unpleasant experience.
Similar coloration raises questions about warning signals and mimicry. Color alone cannot establish mimicry.
New Guinea communities recognized these birds as unpleasant long before laboratory discovery. Traditional knowledge preserved practical information about toxicity. Scientific understanding grew by connecting local knowledge with chemical analysis.
Archived specimens can reveal changes across decades.
Modern methods can measure toxin while preserving valuable material.
Historical samples could track geographic and temporal variation.
Environmental shifts may alter access to toxin-bearing prey.
Chemical defense may weaken if the dietary source disappears. Protecting the bird also means protecting unseen ecological relationships.
One defensive molecule can connect several trophic levels.
Two crucial links remain unresolved.
Scientists still do not know where the molecule is first synthesized.
They also lack a complete resistance mechanism.
The established facts make the remaining mystery even more compelling.
Field evidence shows that the hooded pitahui turns an unknown dietary toxin into one of nature's strangest defenses. This Antarctic fish reaches adulthood without red blood cells, leaving its circulating blood pale and nearly transparent. Its entire evolutionary story unfolded within the isolated, freezing waters surrounding Antarctica. The Antarctic circumpolar current helped isolate southern fishes from warmer oceans for millions of years, supporting Southern Ocean separation. These seas remain cold enough to freeze the body fluids of most unprotected fishes, central to sub-zero seawater exposure. Antifreeze glycoproteins prevent dangerous ice crystals from growing inside many Antarctic nodovoid fishes, central to antifreeze crystal control. These proteins attach to ice and interrupt the repeating structure needed for further growth, supporting ice-binding molecular defense. Cold water slows biochemical reactions, movement, digestion, and nearly every other process inside the animal, supporting cold-slowed biochemistry. A comparatively low metabolic demand reduces the amount of oxygen its tissues must receive each minute, supporting reduced metabolic demand. Nearly every other vertebrate transports oxygen using red cells packed with hemoglobin, while preserving standard vertebrate oxygen transport. Hemoglobin multiplies blood's oxygen capacity by reversibly binding gas molecules inside circulating cells, which affects hemoglobin oxygen binding.
Icefish blood lacks both the red cells and their dominant respiratory pigment, sustaining translucent plasma circulation throughout Antarctic activity.
Under a microscope, its circulation appears strikingly different from ordinary vertebrate blood, which affects erythrocyte-free blood samples. Oxygen must travel physically dissolved within plasma rather than attached to hemoglobin, sustaining plasma-dissolved oxygen throughout Antarctic activity.
That method carries only a small fraction of the oxygen transported by red-blooded relatives, supporting weak oxygen delivery.
Losing hemoglobin reduced blood oxygen capacity by roughly an order of magnitude, with consequences for collapsed carrying capacity. Yet the fish survives because its entire circulatory system compensates for that enormous deficit, supporting whole system compensation. No single adaptation explains life without red blood. Several mechanisms work together continuously, which cold seawater naturally holds more dissolved oxygen than warm seawater, compensated by cold water oxygen abundance inside Antarctic tissues. Abundant environmental oxygen improves diffusion across the gills, despite weak transport inside the blood, which affects gill diffusion efficiency.
The Antarctic environment made a normally catastrophic blood defect temporarily survivable.
Adult crocodile icefishes are the only known vertebrates completely lacking circulating red blood cells, central to adult erythrocyte absence. Their developmental program no longer produces the mature oxygen-carrying cells typical of vertebrates, supporting arrested blood cell development. Genomic evidence records the breakdown of genes required for functional hemoglobin, compensated by globin gene degeneration inside Antarctic tissues. Key globin genes were deleted or reduced to non-functional genomic remnants, compensated by non-functional genomic remnants inside Antarctic tissues. Globin loss occurred after the icefish lineage separated from hemoglobin-producing notothenioids, while preserving post-divergence hemoglobin loss. Modern white-blooded icefishes inherited the condition from a common ancestor, compensated by shared ancestral deletion inside Antarctic tissues. The family later diversified while retaining its unusual blood physiology, compensated by icefish family diversification inside Antarctic tissues. Myoglobin followed a different evolutionary path and disappeared independently in several icefish species, which affects independent myoglobin loss. Where present, myoglobin helps buffer oxygen inside constantly active heart cells, revealing cardiac oxygen buffering under stable cold conditions. Some icefish hearts function without either hemoglobin or cardiac myoglobin, sustaining myoglobin-free heart function throughout Antarctic activity. Independent losses demonstrate that similar physiology evolved repeatedly within the family, compensated by repeated evolutionary loss inside Antarctic tissues.
Scientists still debate whether hemoglobin loss was advantageous or merely survivable under extreme cold.
Removing red cells made blood easier to pump while severely weakening oxygen transport, which affects viscosity-capacity trade-offs. Lower viscosity reduces resistance within the circulation, partly offset through low-resistance plasma flow across cold tissues. Greater cardiac output partly offset the small amount of oxygen carried per unit blood, which affects elevated cardiac output. Wide vessels allow high-volume flow without excessive pressure, partly offset through wide vessel circulation across cold tissues. Dense capillary networks shorten the distance between plasma oxygen and tissue cells, tied closely to dense capillary architecture. Mitochondrial abundance increases the cellular surface available for oxygen use, partly offset through mitochondrial oxygen use across cold tissues. Icefish mitochondria also display unusual density, size, and internal organization, partly offset through unusual mitochondrial morphology across cold tissues.
Hemoglobin loss reshaped anatomy from genes to vessels, heart, and cellular energy machinery, especially through genome-to-heart compensation.
Every delivered oxygen molecule begins as gas dissolved within the surrounding seawater, while preserving gill plasma oxygen transfer. Lower temperature increases the amount of oxygen water can hold, compensated by temperature-dependent oxygen solubility inside Antarctic tissues. Polar conditions therefore maximize the oxygen available for passive uptake, compensated by polar passive uptake inside Antarctic tissues. Thin exchange surfaces reduce the distance oxygen must diffuse, partly offset through thin membrane diffusion across cold tissues. No cellular carrier captures the gas after it enters circulation, revealing carrier-free circulation under stable cold conditions.
Carrying more plasma increases the total dissolved oxygen moving through the animal, sustaining expanded blood volume throughout Antarctic activity. Icefishes maintain substantially greater blood volume than many related fishes, partly offset through high plasma volume across cold tissues.
Cell-free blood flows with comparatively low resistance, partly offset through low cellular crowding across cold tissues.
High vascular density increases the area available for oxygen delivery, compensated by vascular oxygen delivery area inside Antarctic tissues. Short diffusion distances compensate partly for low oxygen concentration, partly offset through short tissue diffusion across cold tissues. Delivery succeeds through broad distribution rather than concentrated oxygen cargo, partly offset through distributed capillary delivery across cold tissues. Behavior also keeps oxygen demand within the circulation's restricted capacity, partly offset through behavioral oxygen conservation across cold tissues. Intense activity can challenge a system with little oxygen reserve, partly offset through burst swimming limitation across cold tissues. Icefishes generally possess limited scope for prolonged high-performance swimming, partly offset through low aerobic scope across cold tissues. Warmer conditions raise metabolic demand while lowering water's oxygen content, compensated by warming-driven oxygen mismatch inside Antarctic tissues. Temperature therefore compresses the animal's physiological safety margin from both directions, compensated by shrinking physiological margin inside Antarctic tissues. Its unusual blood works best within the environment where it evolved, compensated by stable sub-zero performance inside Antarctic tissues.
Depth and local circulation can provide relatively stable thermal conditions, compensated by deep water thermal refuge inside Antarctic tissues.
Shallow populations may encounter faster environmental change through shallow water exposure, limiting stress while sustaining water exposure.
Environmental oxygen and cardiovascular compensation remain inseparable parts of the same survival system, which affects environment circulation coupling. Icefish hearts are enlarged relative to those of similarly sized red-blooded relatives, which affects enlarged ventricular anatomy. Each beat moves an unusually large volume through the circulation, supporting high stroke volume throughout Antarctic cardiac activity. High stroke volume raises cardiac output without requiring extremely rapid heart rates, with consequences for cardiac output compensation. Cold cardiac muscle contracts slowly and depends on specialized cellular machinery, compensated by cold cardiac machinery inside Antarctic tissues. Mitochondrial density supports aerobic energy production inside the oversized heart, partly offset through heart mitochondrial density across cold tissues. Dense vessels keep oxygen pathways short within cardiac tissue, partly offset through short cardiac diffusion across cold tissues. Some species add myoglobin storage within the heart, partly offset through retained cardiac myoglobin across cold tissues. Other species survive after losing cardiac myoglobin independently, partly offset through independent myoglobin absence across cold tissues. Different lineages reach similar physiological outcomes through distinct genetic changes, partly offset through convergent cardiovascular outcomes across cold tissues.
Elevated nitric oxide signaling may help promote wide vessels and vascular growth, tied closely to nitric oxide signaling.
Nitric oxide regulates both blood flow and formation of new vessels, compensated by angiogenic vessel growth inside Antarctic tissues.
Long-term signaling can reshape anatomy throughout growth, through developmental compensation.
Vascular differences emerge as the animal develops, through heart growth trajectories, limiting stress while sustaining heart growth trajectories in Antarctica.
Static signals may transform a blood defect into a stable body plan, sustaining homeostatic circulatory remodeling throughout Antarctic activity. Low viscosity makes large volume pumping mechanically easier, partly offset through viscosity-enabled high flow across cold tissues. Tips. Cardiovascular compensation still requires substantial energy. Through energetic compensation costs, limiting
the fish pays for weak blood with a larger circulatory apparatus, revealing large circulatory investment under stable cold conditions. Warmer water can push that enlarged system toward its performance ceiling, sustaining thermal cardiac limits throughout Antarctic activity. Cardiac capacity may limit acute warming tolerance in some species, partly offset through cardiac performance ceiling across cold tissues.
Every compensation traces back to an ancient loss recorded in the genome, with consequences for integrated polar physiology. Their closest red-blooded relatives still retain that ancestral system, partly offset through retained relative physiology across cold tissues.
A major deletion destroyed essential hemoglobin coating material, partly offset through beta-globin deletion across cold tissues. Additional remnants preserve a molecular fossil of the lost pathway, compensated by alpha-globin genomic fossil inside Antarctic tissues.
Once a protein was no longer required, disabling mutations accumulated more freely, central to post-loss mutation accumulation.
Blood physiology changed far beyond the globin genes themselves, partly offset through reduced iron physiology across cold tissues.
White blood contains dramatically less iron bound in respiratory pigment, compensated by iron-poor white blood inside Antarctic tissues. The absence of red cells lowers viscosity substantially, partly offset through erythrocyte-free viscosity across cold tissues.
Lower viscosity may have softened the damage without making hemoglobin loss beneficial, sustaining evolutionary trade-off interpretation throughout Antarctic activity. Environmental conditions created a rare window in which the mutation could persist, sustaining cold ancestral tolerance throughout Antarctic activity.
Similar gene loss would be far less tolerable in warmer, oxygen-poor seas, which affects warm water incompatibility.
Red-blooded relatives often retain greater capacity for oxygen transport during thermal stress, which affects relative thermal capacity. Extreme specialization can increase success in stable environments while reducing resilience to rapid change. Supporting specialization genome studies reveal additional adaptations beyond blood and circulation, partly offset through antioxidant gene expansion across cold tissues. Oxygen-rich water and abundant mitochondria can increase oxidative stress, partly offset through mitochondrial oxidative pressure across cold tissues. Strong antioxidant and repair systems limit damage from reactive molecules, partly offset through macromolecular repair defense across cold tissues. Icefish survival depends on a broader Antarctic molecular toolkit, partly offset through broader cold adaptation across cold tissues. Traits optimized for constant cold may become liabilities during warming, partly offset through warming-created mismatch across cold tissues.
Climate change modifies several ecological pressures simultaneously through multi-factor climate pressure.
Risk will vary among species, locations, depths, and life stages, partly offset through species-specific exposure across cold tissues. Warm water holds less dissolved oxygen while animal metabolism accelerates, partly offset through warming oxygen decline across cold tissues. Icefish face simultaneous reductions in supply and increases in demand, compensated by aerobic supply-demand compression inside Antarctic tissues.
Limited oxygen carrying capacity may bring some ice fishes to their ceilings sooner, tied closely to earlier performance ceilings.
Thermal vulnerability is not identical across the family, partly offset through variable family vulnerability across cold tissues. Behavioral movement may reduce exposure to warming surface layers, partly offset through behavioral thermal refuge across cold tissues. Mutagens. Suitable cold refuges may not exist everywhere. Through geographic restraints, limiting stress, while early life stages may encounter different thermal risks than adults, compensated by larval thermal exposure inside Antarctic tissues. Warming can reshape development before circulatory compensation is fully established, balanced through developmental oxygen stress during polar life. Changing prey fields can indirectly affect icefish energy budgets, partly offset through changing prey networks across cold tissues. Reduced food could make costly cardiovascular compensation harder to maintain, partly offset through cardiovascular energy deficit across cold tissues. Field monitoring is essential for separating short experiments from population trends, with consequences for long-term population monitoring. Collections reveal anatomy, genetics, diet, and physiological condition, partly offset through research specimen collection across cold tissues. Modern tools connect whole animal performance with molecular mechanisms, partly offset through integrated molecular analysis across cold tissues. Comparative studies identify which traits followed hemoglobin loss and which evolved earlier, with consequences for comparative evolutionary mapping. Variation within the family turns icefishes into a natural evolutionary experiment, with consequences for within-family physiological variation.
Their biology offers insights into oxygen transport without providing a direct model for human treatment, supporting biomedical research boundaries.
Genes. Understanding compensation may illuminate general principles of cardiovascular plasticity, partly offset.
Those adaptations remain inseparable from the cold, oxygen-rich sea that made them possible, helping explain Southern Ocean dependence.
The future depends on whether physiology can keep pace with changing Antarctic water, tied closely to future warming pressure.
Antarctic icefish survive without red blood by rebuilding nearly every other part of oxygen delivery, supporting integrated oxygen delivery.
This small lizard escapes predators by diving underwater and repeatedly breathing from a silver bubble attached to its snout.
Water anoles inhabit tropical stream margins, where dense vegetation, wet rocks, and moving water provide immediate underwater refuges. Their streamside habitat places branches, roots, and climbing surfaces directly above pools, channels, and concealed submerged shelter.
Perching close to water lets the animal disappear from danger within seconds, before many terrestrial hunters can react.
Birds, snakes, and other predators create intense pressure for rapid escape behavior along exposed rainforest stream banks. Before diving, the lizard evaluates distance, cover, current, and the nearest safe route beneath the water safely. A sudden leap carries the animal below the surface before most land-based predators can follow or accurately track it. Once submerged, depth, darkness, ripples, and visual distortion make the small reptile surprisingly difficult to locate from above. Strong toes and curved claws help it remain anchored against flowing water while hiding beneath rocks or roots. The extraordinary respiratory behavior begins when air leaves the nostrils and forms a bubble instead of floating away. Hydrophobic scales help exhaled air remain connected to the face, keeping the bubble accessible beside both nostrils. Semi-aquatic anoles possess scale features associated with stronger water repellency and more reliable underwater bubble retention. The nasal bubble may connect with a broader reflective air layer trapped against hydrophobic scales across the head. Air moves through the nostrils while the mouth stays closed, preserving a compact respiratory circuit beneath the surface.
The lizard inhales from the bubble, exhales back into it, then repeats the same cycle during submersion. Every breathing cycle changes the bubble's composition because metabolism consumes oxygen while tissues continuously produce carbon dioxide.
Oxygen follows the ordinary vertebrate route,
from the lungs across thin tissue and into the circulating bloodstream.
Rebreathing extends the usefulness of existing lung air, rather than creating an entirely new respiratory organ underwater. Measured oxygen decline inside the bubble confirms that its trapped air actively supports the submerged animal's respiration. Repeated inhalation lets the lizard reuse air that would otherwise escape immediately toward the stream surface after exhalation. Recycling stretches the useful duration of a limited air store, while the animal remains concealed during a defensive dive. Each extra underwater minute increases the chance that a searching predator leaves before the lizard must resurface. Staying below the surface allows currents, shadows, and ripples to erase the original location of the sudden escape. Recorded submersions vary greatly because body size, stress, temperature, habitat structure, and individual decisions all influence duration. Some semi-aquatic animals have remained underwater for roughly 8 minutes, although most defensive dives are considerably shorter. A maximum observation reveals biological capacity, not the routine dive length of every animal under natural conditions.
Body size, water temperature, current speed, and disturbance can all change how long a submerged lizard remains hidden.
Smaller and larger animals carry different lung volumes, metabolic demands, and bubble surface areas relative to body mass. Remaining nearly motionless lowers oxygen consumption, allowing the bubble's limited respiratory value to last substantially longer under threat. Sheltered positions behind roots and stones reduce physical effort while hiding the lizard from movement above the water. Calm pockets behind obstacles demand less energy than exposed current, where the animal must constantly resist downstream force.
Moderate flow can refresh gases around the bubble without tearing the delicate air layer away from the snout.
Carbon dioxide readily dissolves into water, creating a possible route for respiratory waste to leave the bubble.
Because carbon dioxide is highly soluble, surrounding water may absorb it faster than oxygen disappears from the reservoir. An exposed bubble water boundary could therefore remain useful longer than a completely sealed pocket containing the same air.
Metabolic carbon dioxide travels from active tissues through blood toward the lungs, even while the lizard remains submerged. Exhaled carbon dioxide enters the nasal bubble, where some molecules may diffuse outward into the passing stream. Carbon dioxide removal is physiologically plausible, but recycled stored air remains the mechanism supported most directly by experiments.
Underwater rebreathing combines ordinary lungs, hydrophobic skin, trapped air, and a dynamic gas boundary beside the nostrils. The respiratory loop begins with oxygen already stored inside air, carried beneath the surface during the initial dive.
Bubble oxygen concentration falls predictably while a living lizard continues breathing repeatedly from the attached reservoir underwater.
Researchers compared that decline with artificial bubbles held underwater under similar physical conditions, but without breathing animals.
Control bubbles retained oxygen more consistently, demonstrating that biological respiration caused the stronger decline seen with lizards. Those measurements confirmed that the nasal bubble functions as an extension of the animal's existing pulmonary air supply. Oxygen enters the lungs during inhalation, while mostly unused nitrogen returns to the bubble during the following exhalation. As oxygen decreases, nitrogen occupies a greater fraction of the remaining bubble, and the respiratory mixture becomes progressively poorer. Eventually, available oxygen falls below a useful level and contributes to the lizard's decision to return toward air. The animal releases its underwater grip, swims upward, and breaks the surface to obtain a completely fresh breath. At the surface, the depleted bubble merges with open air, while the lungs refill within a single respiratory cycle. After climbing onto a nearby rock, the lizard restores normal oxygen delivery while monitoring the streamside environment for danger. Heart rate, breathing, and movement gradually return toward ordinary levels as the animal recovers between defensive dives.
Another predator may appear before recovery finishes, forcing the lizard to repeat its underwater escape under greater physiological pressure.
A second dive creates another nasal bubble immediately, showing that the behavior is repeatable whenever danger returns.
Every resurfacing restores a fresh gas mixture, replacing the oxygen-poor reservoir produced during the previous submerged interval. Across a single day, repeated cycles of diving, rebreathing, resurfacing, and recovery may occur around the same stream refuge.
Underwater movement may also support exploration or foraging, although predator avoidance remains the best established ecological function. Submerged stones, roots, and leaf pockets contain potential prey, as well as concealed resting places away from terrestrial hunters. The lizard balances underwater opportunity against limited oxygen, physical effort, and the continuing need to return for atmospheric air.
Scientists tested the bubble's actual respiratory value by experimentally preventing its normal formation during controlled defensive dives.
Hatched lizards entered identical water chambers, differing mainly in whether the familiar nasal air bubble remained available. Control animals preserved their ordinary hydrophobic air interface and continued cycling exhaled gas beside the nostrils underwater. A temporary surface treatment disrupted bubble attachment without changing the chamber's water temperature, depth, or overall handling conditions. After treatment, exhaled bubbles detached from the face and floated upward before the lizard could inhale them again. Those animals relied mainly on their initial lung supply, because reusable gas no longer remained beside the nostrils. Lizards retaining normal bubbles stayed submerged substantially longer than animals whose external air interface had been disrupted. In that experiment, ordinary rebreathing increased average dive duration by approximately 32% under otherwise matched conditions.
Comparable body sizes, chambers, and handling procedures strengthen the conclusion that access to the bubble caused the difference.
The result demonstrated that the air pocket performs a functional respiratory role, rather than merely reflecting trapped surface air. However, the experiment did not show permanent underwater breathing, unlimited oxygen renewal, or complete independence from atmospheric air. The bubble remains a finite reservoir, whose volume and gas composition continue changing throughout every submerged breathing cycle. Recycling improves how the initial air supply is used, but it cannot prevent eventual oxygen depletion during long dives. Possible carbon dioxide loss across the bubble surface may further delay the point when resurfacing becomes physiologically necessary. Gas may move in both directions across the interface, although each proposed exchange process requires separate experimental evidence. Confirmed rebreathing, plausible carbon dioxide removal, and emerging oxygen uptake evidence therefore occupy different levels of scientific certainty. Successive studies progressed from behavioral observation toward gas measurement, bubble disruption, and controlled environmental experiments over several years. Natural dives remain essential because laboratory mechanisms evolved to solve ecological problems along predator-filled tropical streams.
In the wild, bubble-bearing lizards hide beneath ledges and roots, while threats continue searching along the bank above.
An extended dive allows danger to move farther away before the concealed animal cautiously returns to the surface. Comparative research revealed that underwater bubble rebreathing appeared independently in several distantly related semi-aquatic anole lineages. Separate evolutionary branches developed remarkably similar diving behavior, despite lacking one recent bubble-breathing common ancestor within Anolis. Streamside species from different American regions encountered comparable combinations of predators, water access, and underwater shelter near flowing water. Recurring ecological pressures repeatedly favored escape strategies that turned nearby streams into temporary defensive refuges during attacks. Across independent lineages, threatened lizards entered water and formed accessible nasal bubbles using similar behavioral sequences under threat. Ordinary anole skin already possessed water-repellent microstructure before specialized underwater rebreathing evolved within semi-aquatic species. Natural selection likely co-opted that existing surface property, giving hydrophobic scales an additional respiratory function underwater. Water repellency may originally support self-cleaning or surface protection, while later becoming useful for stable bubble retention. Semi-aquatic forms maintain more persistent external air films that remain attached during movement beneath the water surface. The accessible air concentrates near the nostrils, allowing repeated inhalation while the lizard keeps its mouth closed underwater. Different species vary in bubble size, attachment area, head shape, and the number of threads within their air layer. Convergent solutions still reflect each species' body dimensions, scale texture, habitat, and preferred underwater hiding behavior underwater. Local stream conditions can determine which bubble geometry and diving strategy produce the greatest survival advantage locally. Fast rocky channels and slow shaded pools impose different costs through current strength, oxygen availability, and concealment. Calm water reduces the energy needed to hold position, though surrounding gases may refresh differently than in strong flow. Water containing more dissolved oxygen creates stronger conditions for oxygen to move toward an attached underwater bubble. Researchers therefore compared dive duration across chambers containing low, intermediate, and high dissolved oxygen concentrations in controlled trials.
Lizards remained submerged longest in the highest oxygen treatment, when water movement was kept relatively low and controlled.
Separate experiments manipulated dissolved oxygen and flow independently, preventing one environmental variable from masking the other's effect.
With oxygen held constant, slower flow treatments also produced longer dives than faster-moving water inside the experimental chambers.
The combination of adequate dissolved oxygen, low physical effort, and a stable bubble suggested an additional respiratory possibility.
A true physical gill would draw some dissolved oxygen from surrounding water into the bubble during submersion.
Aquatic spiders and insects already use air films or bubbles that exchange respiratory gases directly with water. Across such interfaces, oxygen can